Hello, Joshida. Good afternoon. You can see my screen, right? Yeah, you can see my screen, right? You can also write in chat box if you can see my screen. Yes. Should we start or should we wait for like 2, 3 more minutes so that if someone wants to join, they will join? Okay. Okay. We'll start with the lecture. So this is the week 4 lecture and this is a 4-week course. So this is the last lecture for our NPTEL course, which codes for BT78, which is proteins and gel-based proteomics. If I get a chance of getting one extra lecture, I might take that, or else this will be the last lecture. So in the week 4, sir has majorly dealt with two major techniques, that is DIGE and mass spec. DIGE indicating differential gel electrophoresis, so in this specific week, he took 5 lectures, and I've divided those 5 lectures into 5 parts similarly. And in the first lecture of week 4, Sir majorly talked about the basics of DIGE, and then Sir talked about how can we do data analysis in terms of using the specific experiment, that is DIGE itself, and applications of DIGE. Those were the initial 3 lectures belonging to DIGE technique itself. And later 2 lectures were based on mass spec technique. So how exactly mass spec can be used for in order for basically to help to understand proteomics or do proteomic analysis. So it will help. So till DIGE, you can still do certain amount of uh, proteomics analysis, but to make it more accurate, you can use mass spec. In mass spec, also one of the technique is MALDI-TOF or MALDI-TOF. So TOF is basically mass analyzer. MALDI is the ionization source. So that will basically ionize your protein and the protein of your interest. And TOF is the analyzer, so it will basically analyze the protein depending upon mass to charge ratio. That's what Sir talked about in lecture 4, protein identification using MALDI-TOF. And in the later part, Sir talked about how can we do data analysis and certain challenges related to the proteomics. So what I'll be doing in today's lecture, similar to that of initial 3 weeks, I'll be just brushing up what Sir has talked about in all these 5 lectures initially, and then we'll go to question and answers of regarding the same topic. Yes, I hope you all are getting it. Yes. Okay. I'll start with the lecture. Okay, as I said, so I'll be initially talking about these. whatever sir has talked about in the all these 5 lectures, and then we'll deal with question and answers. And if you are having any kind of doubts related to all the 4 weeks lectures as well as today's lecture, we can discuss on that after that, like after doing the question and answer session. Yeah. So what sir talked about in terms of DIGE is, so what is DIGE basically initially to introduce? So it is a method to overcome intrinsic gel-to-gel variability. So in two-dimensional electrophoresis, which we did in our third week, what exactly that is, we have dealt with that specific technique right from sample preparation, how exactly we need to do sample preparation till the end, like how exactly analysis needs to be done using 2D gel electrophoresis. That's what we learned, but there is some gel to gel variability generally seen in terms of 2D gel electrophoresis, but Dye technique will overcome that specific limitations of 2D gel electrophoresis. So that's one of the major advantage of using dyes instead of two-dimensional electrophoresis, and this was method which was first reported by Unlu, one of the scientists. And it is basically a protein labeling and suppression technique. Initially, you will label the proteins. So, whereas in terms of two-dimensional electrophoresis, we are not labeling as such. We are just separating them in 2 dimensions, one in isoelectric focusing and one in SDS page. We are not labeling those proteins. We are just separating them and trying to analyze depending upon their molecular weight and depending upon their isoelectric point. We are trying to analyze which proteins might be present in your sample. But in terms of dyes, we will initially label the samples as well and then do the separation similar to that of 2D gel electrophoresis. But there is labeling part included in terms of 2D dyes technique. And it's an optical detection of proteins with, so this protein labeling is done with fluorescent tags basically, and it will help you to get a linear detection in wide range of proteins. Basically, there are in a sample, there are abundant proteins, there are less concentrated proteins. All of those can be analyzed in terms of by using 2D dyes. So, what is the basic principle of dyes? Is it is commonly used fluorescent protein labeling reagent for dyes. So, certain fluorescent tags are Psi2, Psi3, and Psi5. What are these Psi dye? Basically, they are the ester derivatives of this specific compound, and the size and charge matched is equal to that of labeled samples. They co-migrate within the gel itself. So we also mix these dyes basically, and uh, I'll come to those method how exactly we do this specific technique. So, just giving a detail about what these fluorescent tag dyes are. So, there are 3 different types of these dyes, Psi3, Psi5, and Psi2. And this is a bit of molecular difference. They are almost similar molecular weight, but they have some difference like 580 to 580 and 550. And but they have different excitation and emission wavelength. So this is what the benefit is basically. Although they have a similar molecular weight, their excitation and emission is different. So accordingly, while doing the analysis, the sample in which we added one specific dye, we can then accordingly use certain excitation wavelength and emission wavelength, and we can analyze those specific proteins of the sample itself because of its specific excitation and emission wavelength. So this is the major advantage of using these dyes, and that is the reason why these dyes are used, because they have different excitation and emission wavelength, all three, and they have a similar molecular weight. So they are not even adding to the whole molecular weight while doing the separation technique and stuff. And these certain more advantages, like they are photostable, they are pH insensitive. So if there is a change in pH, it will not affect them, and they are spectrally distinct as well. That's what spectrally distinct means. The excitation and emission is different. So there are 2 labeling methods basically. So as I said, in this technique, we need to do labeling first. So labeling with these specific dyes itself. And in order to label them, there are 2 different techniques, that is minimum labeling and saturation labeling. Generally, minimum labeling is used, but in some cases, saturation labeling is also used. So what does this labeling does is it basically that these specific dyes basically binds with lysine or cysteine. So it is minimum labeling is so it is basically trying to bind with lysine, and it's called lysine labeling. And similarly, charge and size is matched, and no multiple labels can be done. So that's how this specific labeling is done, and it labels around 3% of all proteins. So it doesn't label each and every protein present in the sample. It will label only 3% of all proteins. Similarly, there is cysteine labeling also, wherein charge neutral and size is matched, whatever charge neutral is there, and multiple labels can be done. So that's the difference here. Only here no multiple labels can be done, whereas in this saturation label technique, multiple labels can be done, and it is very sensitive compared to that of minimum labeling. So, why do we do lysine labeling only as we are having, as we learned in our first lecture itself during the basics of amino acids and proteins? So, we have 20 different amino acids, right? And out of those 20 different, why are we choosing lysine only? So, this is the specific reason why we are using lysine. So, lysine is preferred amino acid to label because its frequency is more than 5% in most of the proteins, basically. So because of that, so it's expected average 5%. This is in general distribution of amino acid across the proteins. So that's how amino acids are, and we generally choose lysine labeling because its frequency is more than 5%, and statistically almost every protein has at least one lysine residue. Because of that, so even if it has one lysine residue, that protein will be labeled because that one specific lysine is labeled by this side eyes, basically side 2, side 3, or side 5. So that's the reason lysine labeling is done, and whereas cysteine is less than 2% in most of the proteins, so that helps in saturation labeling. So in terms of lysine labeling technique, so cyanine dye react with the primary amine. So how does the reaction exactly happen? So this is how the reaction is. So there is the primary amino group present, wherein this dye comes and binds. So this is the protein, this is the dye, and this specific dye comes and binds with this amino group here at certain pH. If we when we So basically after adding these two, we incubate for around 30 minutes, and we use a certain buffer which has a pH 8.5, and when we do this specific incubation at 0 degrees Celsius, that's how the binding happens, and that's how the protein gets labeled. So it reacts with the primary amine group of the target protein, and N-terminal alpha amino and lysine EM epsilon amino groups, and nucleophilic substitution happens. So this reaction by which it is getting attached, it's called as nucleophilic substitution. And so along with this primary amino group, it also binds with all available lysine labeling would create very hydrophobic proteins. So minimal labeling. So if all available lysines are labeled, then it will create very hydrophobic protein. So that's the reason minimal labeling, only 3% of the whole proteome is labeled. Because if you try and label everything, it will become hydrophobic in nature. And so this is how the structure of a dye is, and it comprises of positively charged side dye, which is this charge is positively dye, and it has a linker. This part is called as the linker, and it has a NHS ester reactive group. This is the NHS ester reactive group, which is reacting here, you can see. And this at pH specific 8.5, NHS ester couples with the amino group, and so this amino group is of lysine in proteins and forms covalent. So it is forming covalent amide group, indicating the bind is very much strong. If you have dealt with a bit of chemistry, so you'll understand what how covalent bonds are. Covalent bonds are very strong bonds, basically. So that's how the labeling happens. So this is the first step in terms of Dice technique, that you need to label your proteins. And how exactly labeling happens? So this is how the labeling happens, and lysine is the amino acid which is targeted by the side dyes, and that's how they are labeled. So in terms of lysine, so the amino group of lysine residue is positively charged at neutral or acidic pH. When it goes to basic pH, it becomes uh, uh, negatively charged. So dye is also positively charged, so the net charge on Pi is effectively uncharged. So this is the isoelectric point Pi indicating. So at that specific point, effectively it is uncharged because dye is positively charged and amino group is also positively charged. So the net charge on Pi is effectively uncharged. Similarly, these two dyes, Psi3 and Psi5, are two spectrally resolvable dyes. So spectrally resolvable indicating Psi3 and Psi5. So it is 540 and 620. So they are spectrally resolvable dyes, and as well as their molecular weight is similar. and and because they have matched for mass and charge as well. That's what I was talking about. So how does the sample preparation happen? So basically you will reconstitute in DMF, which is dimethyl formamide. And there is, so as I said, there is a lysis buffer which has a pH of 8.5. That lysis buffer has these following components, which is stress, urea, thiourea, chaff. We all dealt with this in terms of when we were talking about two-dimensional electrophoresis, and we did talk about how exactly urea affects, how exactly this chaff affects the lysis exactly. So we talked about that in two-dimensional electrophoresis. And so no IPG buffers or carrier ampholytes are required in terms of this. Only these are required when you are doing dyes technique. And around 50 microgram of protein is labeled with, and the concentration of dye which you add is around 400 picomoles. And these reactions are quenched with excess of primary amine, which is free lysine, and 1 microliter of lysine is added. So basically, this becomes a little higher concentration. So a bit of lysine is also provided so that each and everything, so that with the excess of primary amine is added basically to the reaction for it, so that quenching becomes much more appropriate. Along with that, we, in terms of dyes technique, we require an internal standard. So what is that internal standard? It can be of different ways. One internal standard can be pooling of each and every sample, which is called as pooled internal standard. In other words, it is also called as QC in terms of quality control. So this internal standard, can you can also call it as QC when you are pooling everything. So if you, so QC is something. So whatever after proteomics, whatever proteins or peptide bonds you will see in this particular pooled internal control, only those you will consider, only those peptides will be considered in these samples, because this is having each and every peptide involved from all the samples. So if there is any certain contamination in this and it gives certain different peak itself, which is not in the pooled internal control, we'll not consider that. So that's the reason internal standard is used. So pooled internal standard preparation by combining aliquots of equal amounts of proteins from each and every sample. So that specific thing is called as pooled internal control. And there are certain internal standard advantages. So I already told one of the advantage here, but there are, just to note down a few of them. So every protein in population appears on each gel. So if there is an internal standard, so in this internal standard, so all these proteins will appear, right? So every protein in a population appears on each gel, and gel to gel variation reduces because of this. And gel to gel match and comparison becomes much more easier because we have an internal standard. And a reference point for every protein species on each gel in the experiment is obtained, because a reference point will be there because in internal standard everything is there. And automated accurate spot statistics can be performed using this. So these are certain advantages if you use internal standard. So that is sample one, sample two, and an internal standard. So this will have all both the samples together. So in terms of labeling, we generally label samples with Psi3 and Psi5, and we generally label internal standard with Psi2 dye. Because these two are having similar molecular weight. This has a bit different molecular weight, and this is how so basically for samples, maybe for control samples and disease samples, you can divide accordingly and label them. So when you label them with Psi3 and Psi5 and internal standard with Psi2, after labeling your protein samples, you will add those to the You'll basically mix all three of them, and once you do the mixing of all three of it, you will do the same 2D dimension, 2D electrophoresis, which is two-dimensional gel electrophoresis. The similar thing which we learned in our third week lecture. We'll do each and every step after this. So basically, the initial step where we didn't do labeling in terms of 2D electrophoresis. Here we'll be labeling first, and then we'll be doing the isoelectric focusing, similar to that of what we did with two-dimensional electrophoresis, and then we will run the gel after equilibration. So we exactly did do about how exactly DDT will affect the sample, then iodoacetamide will affect the sample. We did all of this in our third week of the lecture. And if you have missed that lecture, I have added the recording in the YouTube. You can watch that so that your concepts are cleared in terms of two-dimensional electrophoresis. So, uh, so basically, after labeling, you will run the isoelectric focusing, and you will also run SDS page. So this is basically your two-dimensional electrophoresis. So once that is done, so this was the first part of the lecture what sir talked about. So just to revise whatever we did. So so these fluorescent dyes are photostable, water soluble, pH insensitive, and these dyes are highly sensitive. Similarly, samples are pre-labeled with one of the dyes and multiplexed within the same gel, and along with an internal standard is required, which provides unmatched statistical confidence. So this is the absorption emission wavelength just to have all three together, and they have different color also, like green, orange and red. So this depends upon the emission wavelength basically, and their molecular weight is also different. Yeah. So that was the first lecture what Sir majorly dealt with, and this is the second part of the lecture. I hope you all are getting it. Hi Aditi. People are coming and leaving. I can see around three, four people have joined the meeting and left the meeting. Yeah. So we'll go ahead. So that was the first part of the lecture which Sir talked about in week 4, lecture 5, all those 5 lectures. And coming to the next part 2 of the week 4 lecture. So in this, Sir majorly talked about how we can use the software basically with one of the professor from GE Healthcare. So I'll not be talking about how exactly the softwares can be used, but I'll be talking about how basically 2D digest performed, what are the benefits of it. So after doing the experiment, how we use the software, what Sir talked about in this lecture, you all can go through that specific lecture. So in terms of this, so image, what Third discussed about in the second part. So he did a bit of image analysis using a software. So that software name is Decider. So once you run the sample, how Decider helps you to analyze the sample. That's what uh, Sir discussed in the part 2 lecture. And there was pairwise analysis between test and control samples. So this pairwise analysis indicating the statistical analysis t-test, and there was analysis between multiple samples belonging to two different groups using biological variation analysis module of the software. So this whole part was actually uh, taught by Sir and one of the professor in two different lectures. That is part one, part two, and part three. So I'll be just giving a gist of the whole two lectures. So what are the certain benefits of using internal standard? So as we already discussed, the internal standard is generally the labeling with Sci2 dye and three images indicating three different dyes. So one sample, one dye, the other sample, another dye, and internal standard, the second dye. So there are three images can be obtained from a single gel itself because we have done the mixture, right? So if you remember while doing the sample preparation, so this sample one we only be visualized when you are using certain excitation and emission wavelength. Similarly, if you take an image with different excitation emission, you will get Psi3 labeled sample proteins. And if you do this excitation, this emission, you will get Psi5 labeled. So basically, with one gel itself, you can get three images because you have mixed these three sample which were having different dyes basically. And after mixing, you have just loaded that specific sample. And while taking an image, while doing the analysis, you took three images from single gel itself, and that image can be taken from one of the scanner called a Typhoon scanner. Then this, once you take the image, then you can take those specific images to Decider software for image analysis. And there are different types of analysis, DIA analysis, which is data independent analysis, wherein which minimizes gel to gel variations, and there is biological variation analysis as well, BVA, which minimizes biological variation. So depending upon your need, what your approach is or what your aim and objectives are, depending upon the experiment, you can accordingly choose DIA or BVA. And so basically, BVA can be further analyzed using statistical tools in EDA. So EDA is also a software. It uses stringent statistical tools like differential expression analysis, principal component analysis, pattern analysis, etc. So now how the labeling happens, so it is explained in detail. For cell surface proteins, labeling in intact cells. For cellular proteins, labeling in lyse cells. So how do you do labeling depending upon your interest? So if it is a cell surface protein, so you don't have to lyse the cell. So while adding this specific buffer, where were we? So this lysis buffer won't be added if you are trying to check the intact cells. Basically trying to check the proteins which are on the surface. So then you will not have this specific lysis. All these components will not be added, which helps in lysis. You'll just have a buffer which has 8.5 pH, and then you will add the dye if you are trying to check the surface proteins. But if you are trying to check the cellular proteins which are inside the cell, then you need to lyse the cell and then check whether those proteins are present. And then cells are incubated with those specific dyes for 20 minutes in dark. And the unreacted dye is quenched with 10 millimolar lysine. That's what we were talking about. The an extra lysine is added so that whatever dye is left, which were not able to contact with the protein, that is the unreacted dye gets also quenched with lysine, and it is easier to remove them. Then same step as that of two-dimensional electrophoresis were done, like that is isoelectric focusing and SDS page. And after that, once you get the gel, that gel is the image is taken using Typhoon scanner, and the analysis can be done using Decider software. So there are certain formats you need to use in terms of when we are taking the image. It is either TIFF image or .gel format. So you need to upload the gel images in these softwares only. Then only that Decider will take those specific images. And then the changes are made with the Sci2 images are extrapolated to Psi3 and Psi5 because Psi2 has internal standard. So this will have each and everything, and they are extrapolated to Psi3 and Psi5, sample 1 and sample 2. Then if you are doing DIA type of analysis, data independent analysis, then for analysis of one set of Psi2, Psi3 and Psi5 images, it will give a 3D view and maximum slope and maximum volume for all the spots can be done in terms of using DIA type of analysis. Then the same data analysis were continued in the other lecture. So this is how the images are obtained. So this is the overall view. So sample one, you are doing Psi 3. As I said, standard is generally internal standard, which is pooling of these two. So sample one and sample two, which are pooled together. That is generally labeled with Psi 2 itself. And the other sample is labeled with Psi 5. When you are mixing all of them, then you are doing the isoelectric focusing and SDS page. And once you do that page, so now while taking an images, you will do different excitation emission images, and accordingly you can take the images for Psi 3, Psi 2 as well as Psi 5. So if you see in Psi 3 and Psi 5, there will be some different proteins. If you see, compare these two images, so this protein is not present here. So indicating that this sample 2 didn't had that specific protein, whereas sample 1 had that specific protein. But if you look at the internal standard thing, which were pooled of these two together, you will see each and every protein present in this as well as this. Right? For example, this protein is not present here, but whereas this protein is present here. So there are certain proteins which were present in sample 2. There are certain proteins which were present in sample 1. But internal standard will have both of them because it was a mixture. So this is basically giving you that statistical advantages to make sure that whatever you did was accurate. So all those advantages we were talking about, that becomes accurate when you are trying to understand these three images. So that's what the overview is. So he tried to give you an example, and so in that specific second lecture, sir majorly talked about how to do those analysis by clicking. So it is easy to use specific software. You all can go through that specific video in order to understand that. It's how exactly software can be used once you get the image by using Typhoon scanner. So, sir tried to give a realistic picture of how exactly then samples are connected, the samples are divided, and how an experiment can be performed basically using two-dimensional electrophoresis. Sorry, using 2D differential electrophoresis DICE technique. So initially, for example, sir gave one of the example, like for example, blood samples are collected, then their serum is separated. So multiple patients, multiple serum samples will be there. Then multiple gels will be run on those samples. So you are collecting the blood samples, then you are separating plasma and serum differently, and only serum samples are uh, collected. Depending upon your experiment, you can do anything basically. And so in serum, there will be certain amount of abundant proteins which we generally know. So those abundant proteins are initially removed, and uh, by using certain chromatographic technique. Once that is done, then you will take the remaining proteins which are present in the sample, and those can be divided into control sample, test samples, and you will pool each and every sample with equal amount and make one of the internal standard sample as well, as we were talking about in the initial part of the lecture. Once you do that, so all the samples will be run on the gel. So there are several 2D gels will run in order to analyze all the patient samples. So these are different patients. Certain are control patients, certain are test patients. Basically, they might be having certain disease. Whatever we are trying to predict. This is just an example of an overview of it, basically. Then those control samples are labeled with Psi 3, test samples are labeled with Psi 5, and internal standard is labeled with Psi 2, similarly mixed, and you will run the gel. Then you will take this is Typhoon scanner, where which helps you to take the dye image, and at different specific excitation emission you will get the image. This is how the image will be, and so this sir this is just an overview of whatever we did till now. You will have the samples, you will do the two-dim isoelectric focusing, you will run the SDS page. Once you run the SDS page, this image, so as you are already mixed, make the sample preparation with all three dyes. So here itself, all three samples, multiple samples can be run together. Whereas in terms of two-dimensional electrophoresis, only one sample can be run at a time. Whereas here, all three samples are run in one go itself because we are mixing it and then running, and all three can be visualized separately because they have been labeled separately, right? So that's the reason even after mixing, we can actually visualize them separately using Dice technique because there is a labeling which was involved initially. Whereas in two-dimensional electrophoresis, there is no labeling as such. So because of that, only one sample at a time can be run using two-dimensional electrophoresis. In DICE, multiple samples can be run. So once you have the image, so this is how the images, real images look, and you can do the analysis, and by analysis you can understand which proteins are overexpressed, which proteins are not present in one of the sample, control sample, and all those control and disease samples proteins should be present in your internal standard. Only then you will consider those proteins. Then in the software itself, you will try and understand whether how dense it is or how not so dense it is. So depending upon that area, so it helps you to understand the three-dimensional protein expression profiles in the software, which we call about decider software. So that's how decider software will help you to understand each and every protein, like if it is overexpressed or if it is present or not present, if the protein is present, what kind of protein it might be depending upon its molecular weight, etc. So that's how all those analysis can be done using this software. So to just to give a recap, so 2DE as well as diets can be planned depending on the objective of the study. So if you just want to have one specific sample and do understand what proteins might be present or what is getting overexpressed or what is getting uh, uh, like not getting overexpressed, but it is getting lowered as compared to that of the control samples. You can do 2D, but so basically, depending upon your objective and study, you can accordingly choose which experiment you need to perform. So DIGE is extremely valuable tool for analysis for large number of samples because it can run multiple samples at a go itself. And as we said, it overcomes the problem of gel to gel variation, and you can have a control and test sample can be differentially labeled using sign-in dyes and can be run on a single gel. That's what we are discussing. So then, sir, try to give certain case study examples. So in order to check what applications dyes is having, I hope you all are getting it till now. Like before the application, how exactly dyes perform. Yes, should we go ahead with the application part? Yes, Aditi. Just a minute. Okay. So moving ahead, sir, gave one of the application perspective of how DIGE can be used and how DIGE technique will actually help you to determine certain biomarkers related to certain diseases. So I just gave an example of one of the study or one of the article which you can go through it, but how exactly they have used DIGE technique in order to understand. So basically, they tried to understand certain serum markers for progression of prostate cancer. So how exactly they collected the sample? How exactly they did plan their experiment using Deitch technique, and then how did they do analysis, and how did they confirm whether these are certain proteins which might be responsible or can act as a marker for progression of prostate cancer. So what is prostate cancer? Basically, it happens to males generally, and so here the objective was to identify serum markers depicting progression of prostate cancer using Deitch technique. So this is prostate specific antigen, a general antigen which is generally present in higher amount when if a person is suffering from prostate cancer. So that was the main protein you are looking for when you are doing Dutch technique. So you have taken one of the prostate cancer patient, their sample, their serum sample. Then there is a healthy control sample. You will mix them. You will do the proteomic analysis. There are people who have done using tissue samples as well. So this is one specific protein which people try to check, and whatever diet methodology we talked about that can be performed, and diagnostic markers for early prostate cancer can be identified using this technique. But there are certain discussions going on related to that. There's if a prostate specific antigen can be the actual biomarker, because we don't exactly know, because in healthy controls also sometimes that is present. Basically, the one which is having benign cancer and not the metastatic cancer, even those are having a bit of high amount of PSA, and that's the reason it is not that accurate enough to understand whether the progression of prostate cancer is happening or not. So, because of that specific reason, they did one of the experiment to understand what other markers can be present in order to understand the progression of prostate cancer. So, as I said, in the serum there are certain abundant proteins. So, if you can see the high molecular weight proteins, which so basically the one which are abundant. So initially, uh, gel-based chromatography technique is performed, wherein injection of sample, then there is size separation, then we do elution. While doing elution, the larger will go out first and the smaller will come out later. So that now you know that the abundant proteins can be removed, and only the proteins which are essential, like here, for example, PSA, or they basically trying to find if there are any other serum markers which can be found out. Then they collected n number of different samples, and they divided those samples, like the disease sample itself, in two different parts, cohort 1 and cohort 2. And cohort 1 had a Gleason score of 5, and cohort 2 had a Gleason score of 7. So what is Gleason score is basically depending upon how early or later stage prostate cancer those patients were. So depending upon that, they are given certain score, and that depending upon that, they have divided them into two different groups depending upon their score basically, and they have run the sample, did isoelectric focusing, then they did the SDS page, and similarly to that of what we discussed, how exactly digest performed, they performed that. And once they performed that, they did a bit of mass spec analysis also. We'll talk about this in the coming slides. So differential protein expression. Once they found that there are certain, so they have run it in two different gels. These two, uh, two different dyes were basically added. This is sample one. This is sample two of different cohorts, and you can see, you can accordingly visualize there are certain proteins which are present in the certain Gleason score patient, whereas certain were not present. If you can compare these two, there are certain proteins which are present here, but they are not present here. And accordingly, one specific protein can be digested. For example, if you want to find whether what is this protein which is not present here, you will do the digestion of this specific spot and do the mass spec analysis. So how exactly those digestion is done and how exactly mass spec analysis is done, we'll talk about that. Then, so in that paper itself, they basically found that these were certain specific pigment epithelial derived factor or zinc alpha 2 glycoprotein. These two certain proteins were downregulated or upregulated accordingly, and so they found these were the certain proteins which can potentially act as the biomarkers. So one of the protein like pigment epithelial derived factor, this one which was downregulated in the uh, patients. So if you can see in the Gleason 5 patients, it was higher, whereas in Gleason 7 score 7 patients, it was lower. If you can see the difference. So how did they confirm this specific uh, variation? Or so here, by doing the mass spec analysis, they found that these were downregulated, these were upregulated, these were certain proteins which were downregulated, and these are the certain proteins which were upregulated. So they found that. So now they want to confirm how much upregulated and how much downregulated, because in mass spec analysis you can just find that it is either upregulated or downregulated. So how much exactly? So they tried to find that using ELISA technique. So using ELISA, so the specific antibody is used. So if it's so your protein of interest is the antigen, and that specific antibody is used. So if in your sample this is present, maximum amount of binding will happen, and then the secondary antibody is added, which is tagged to certain which is tagged to certain enzyme, which is tagged to certain enzyme, and then the substrate is added. And when you add that, it the intensity will change depending upon the amount of binding happened in the first step. So that's how you can find if it had a higher amount of PEDF or it had lower amount of PEDF. And that's what they found. So these patients had higher PEDF, whereas these patients had lower PEDF. So indicating that if it is continuously getting lowered, the PEDF proteins, you can say that the prognosis of or the progression of prostate cancer is happening. Similarly, zinc alpha 2 glycoprotein was also checked, and it was found that it was upregulated a bit compared to Gleason 5 score patients. So that's how they confirmed that. So did understand that it got upregulated or downregulated, but here they found that how much it was upregulated or downregulated by using ELISA as a technique. You can also use Western blot as one of the technique to prove or to validate if these proteins are present for upregulation or downregulation. Then there is also one more technique like immunohistochemistry, similar kind of technique. So this also, so in the tissue sample itself, you can find whether those specific proteins are present or not. So if the binding becomes much more easier, then the Gleason it is higher or it is present in lower amount or it is present in higher amount depending upon whether it is down-regulating or up-regulating. Similarly, this technique can also be used to understand, to validate basically. So that's how they validated in this specific paper that PEDF and zinc alpha 2 glycoprotein are responsible, can potentially act as a biomarker or a marker for progression of prostate cancer. So that's so basically confirmed by differential expression of PEDF and zinc, ZAC by various validation technique, that was immunohistochemistry and ELISA. So these serum markers reflect the pathological grade and stage are beneficial for identification of appropriate treatment strategy. So depending upon that level, we can understand how it has been progressed, the prostate cancer, whether it is in early stage or later stage. So this can be certain other biomarkers. So that's how this technique will help you, and that will help us to understand these are certain biomarkers which can be observed or which can be potentially act as a biomarker and will help you to understand the progression of prostate cancer. So that's what this paper talked about. So the to give an application perspective of Dyes technique, sir discussed about this specific paper. Then, so that's what the Dyes was all about. Then we sir went ahead to so so using Dyes itself, we can still understand how proteins can be identified. Then if further confirmation can be done using MS technique or MALDI-TOF, which is one of the MS technique. So in the next lecture, the part 3 of the lecture, so part 4 of the lecture, so talked about MALDI-TOF. So what is MALDI? It is matrix assisted laser desorption ionization technique, and TOF is time of flight. So this is a mass analyzer basically, and this is the ionization source. So I here stands for ionization. So this, and it has a matrix basically, so it's called as matrix assisted laser desorption. So laser will help to ionize, and matrix is also present, so that's how the name is. And before this specific step, there is one step called as in-gel digestion. So sir gave an introduction of what MALDI-TOF is, and then talked about how exactly digestion is done. So as I was talking here, uh, uh, how they did mass spec. So here we'll, in this part of the session, we'll discuss how exactly MALDI-TOF is done. So that's how they did and understood that these were upregulated, these were downregulated, and they further validated using ELISA and immunohistochemistry, and that's how they found these are certain proteins can act as a biomarkers. Yeah. So before going to mass spec, so that's what DIGE is all about. Then, after performing the diet, in order to make your analysis much more accurate and identify the specific protein itself, you can do mass spec analysis. And one of the commonly used mass spec technique for proteomic study is MALDI-TOF, which Sir talked about in the next two lectures. I hope you all are getting it. Yes, yes, Aditya, are you there? You can write in the chat box, or you can unmute yourself and talk. Yeah. So that's what diet is about. Diets about diet technique what sir discussed, and as I said, to make more accurate, we can also do MALDI-TOF for making it for in order to obtain or identify one specific protein accurately. So to give an introduction, what exactly mass spec is? So mass spec is a technique. So it basically it's one of the separation technique again, but it separates depending upon mass to charge ratio. So it's a technique production of charged molecular species. So initially it is ionized, it is given a certain charge, and then the separation happens by magnetic and electric fields. So there is magnetic and electric field because of the presence of quadrupoles, or and because of that there is magnetic and electric field is being created at that specific zone, and based on the mass to charge ratio, they separate. And when they hit the detector, according to what specific peptide might have hit the detector accordingly, depending upon their intensity also, depending upon their mass to charge ratio, that's how the peaks appear. And this is how you get the mass spec data. So to give a general introduction of what mass spec is, so you initially need ionization, and here the ionization which we are using in this technique is MALDI, matrix assisted ionization technique. And after you ionize, you can separate and do mass analysis. And once that is done, the detection and generation of mass spectrum is obtained. This is how the mass spectrum looks. So you have the 2D gel electrophoresis. Now you have the so this is how a typical workflow is. So once you do the DIGE technique and then identification of those proteins. So if you want to identify what is this specific protein, what is this specific protein, or what is this specific protein, or what is this specific protein. You need to cut the gel basically, do the in-gel digestion, and after doing the initial sample preparation for mass spec, after doing this digestion, you will understand what specific protein that protein is, depending upon the spectrum. So each protein will have its own spectrum. So after analyzing the gel, one needs to identify differentially expressed proteins. So that is what CIDER software will help you. So differentially expressed. So what uh, that means? So for example, here this is the protein which is not present, but here it is not present, but here it is present. So these are differentially expressed proteins, right? So that analysis you need to do first. And once you do that, you can just cut that differentially expressed protein and do the mass spec analysis. You don't have to do all the protein. So if this is present here and also present here, you don't have to analyze that specific protein. You need to analyze only those proteins which are differentially expressed using mass spec. Yeah. Sorry. Okay, so that software, so that's how the software looks. And so if you point out one specific spot, then that specific spot will, that's how it will tell you how intense it is, whether it is overexpressed or whether it is not overexpressed. Then accordingly, you will understand. That's how software will help you. So it will establish the identity of proteins. Different types of mass spec platforms can be utilized. So now once you understand that these are certain differential express protein, you will cut that specific protein and do the in-gel digestion, and so that do the mass spec analysis, and for that different mass spec platforms can be utilized. So here we discussed about only one platform, which is MALDI-TOF. So one of the platform for doing this analysis or identifying the protein. So now you know that this is the specific protein which is not present in the control, or something which is not present in the control but present in the patient, or something which is not present in the patient but present in the control. You can also check vice versa. So now those specific proteins can be taken, and so before that, as we are using MALDI-TOF as a technique for analysis, so what exactly MALDI-TOF is? So MALDI is an ionization source, which is matrix assisted laser desorption ionization technique. So there is a sample plate. We'll discuss in deep about how exactly that plate looks, how do you add the samples in the further slides. So this is just an overview of that. So there's a plate, and there are certain small wells in that specific plate. You will add the your after your sample preparation of the in-gel digestion you have done, and you have prepared the sample for mass spec. You'll just load that uh, sample here along with the matrix. So there is a matrix as well. We'll discuss about matrix as well. You will just load that. Then there is a laser desorption. The laser will ionize the samples, and that ionized samples will go through the time of flight region, wherein it will try and analyze the masses. Depending upon mass to charge ratio, they are separated here. So using MALDI as an ionization source, these are certain advantages. Like it requires sample preparation is very easy in terms of this technique. It is more tolerant to salts than that of electron spray ionization. So this is another ionization technique, electron spray ionization, but MALDI is more tolerant to salts and it produces singly charged ions as well. And but one of the disadvantages, it strongly depend on sample preparation method. So what sample preparation method you are using, your MALDI results will differ. So accordingly, only one specific method should be used for all the samples so that it is replicated properly. So MALDI is a soft ionization technique. It involves easy sample preparation. It is more tolerant to salt. It produces singly charged ions, and it provides high throughput platform to basically to identify a protein using MSMS. So that was the ionization source. So this ionization source here, there will be a sample, and that sample will be so like this basically, like this. Just a minute, like this will be present here. So the plate will be going here where your samples are, along with the matrix. Then the laser will come in contact with that, and it will ionize the uh, matrix assisted plate. And at the time of flight region, the those specific ions will be separated depending upon their mass to charge ratio. And once they are separated here, and once they hit the detector, the spectrum will be so this is connected to the computer, and it will like whatever detector is getting the information, it will give the peaks accordingly. So it's a mass analyzer, basically consists of ion acceleration. So here there is an acceleration and focusing optics. So this is the focusing optics, and the flight tube. So this is the flight tube, basically the region you are seeing here. It measures mass to charge ratio of ions based on time, and it takes so basically what based on time it takes for ions to fly in the analyzer. So this is the analyzer. So depending upon the time it takes for the to fly it and strike the detector, depending upon that, it will try and analyze what specific peptide that might be. So if you can see, the lower weighted proteins masses are going faster as compared to that of higher weight one. So there's an equation to find out the time. So this is what the equation is depending upon their mass. So if you can see, the lower mass are going faster, higher mass are going slower, and whichever hits the detector first, you will get the peak of that specific peptide first. If you can see here, initially red, blue, and then the dark blue, and then accordingly those peaks are formed. So it resolves ions based on their mass to charge ratio. It measures mass to charge based on the time it takes to fly in the analyzer and strike the detector. And lighter ions travel faster and strike the detector before the heavier ones. That's what we were talking here. So there are different types of mass analyzers in terms of time of flight mass analyzers. One of it is called as flight tube. Or here, as I said, the lighter ions will travel faster. So once, so this in this flight tube, and there is a detector, and the compared to that of heavier ions, and it can be correlated with mass of the ion itself. There is one more technique in order to increase the path length. There is a reflection mirror which is added to the time of flight instrument itself. So without increasing this length, you can actually increase the path length of those ions moving towards the detector. So here the ions will come from the ion source. The ions will come and first hit the reflection mirror, and once they hit the reflection mirror, then they will go towards the detector. So this is the time which they have extra till they go towards the detector. So there is a reflectron mode as well. Here there is another mode where there is no reflectron present. So this will increase the path length, and it becomes the analysis becomes much more uh, the separation becomes much more accurate here, because here the path length is small, and continuously some specific peptides might hit here the detector, whereas here as the time is more, the detector might get enough time to understand, and the peaks will also be separated in a good way. So as I said, this is the equation which deals with t is the time of flight, m is the mass of the ion, c q is the charge of the ion, and v is the accelerating potential, and l is the length of the tube. So here the length is this much, whereas in this technique the length is this plus this, both of them together. So that's how MALDI-TOF works. So as I said, so just to give an introduction how MALDI-TOF works, then after that, how exactly initial preparation is required in order to perform MALDI-TOF. So for example, you want to detect this specific protein. what is this specific protein you want to understand. So now you'll cut this protein using a clean uh, scalpel, and you will, after cutting that specific spot, you will add it in the lysis buffer which we were talking about. Then you will do trypsin digestion, and what exactly trypsin do? It cuts the C terminal of arginine and lysine. Then the whole protein is digested into certain peptides, basically, like this. And after you have the digested protein, you will try and remove whatever salts, buffers, detergents you have used in order to lyse it. You'll just remove all of them. And now you have the sample. In that sample itself, you will add the matrix as well, as it is matrix associated laser desorption technique, ionization technique. So you need to add the matrix also. We'll talk about what all different matrix are available. And once you have that, then you will do the, so this is how the sample plate looks. You will add the samples here accordingly, and you can place this your plate. So this specific, once the samples are added here, after adding the matrix, we'll incubate and then add the samples here on the 196 well MALDI plate. So you can analyze 196 samples at a go using MALDI. So now you added the samples, and you kept the plate here, inserted the plate, then the laser will come and it will ionize the sample, and it will, the ions will then, depending upon the mass to charge ratio, they will travel towards the flight of tube and hit the detector, and the detector will accordingly give the, as soon as it hits the detector, according to their intensity and the time it took towards, so this is the time basically, time it took to hit the detector, you will accordingly see the peaks of the peptides. So that's how the analysis happens using MALDI-TOF. So this is overall overview of what we discussed. So you're just taking the sample. Uh, so for example, you want to detect this specific protein. You will excise that specific spot. You will destain it and extract the protein out of the gel. Once you have this sample, you'll do the digestion of it. That is trypsin digestion, and trypsin digestion basically lysine and arginine. C terminal is cut. And there is one limitation with trypsin is, so if after uh, arginine or lysine, if there is a proline present after that, then the digestion generally doesn't happen. And if there is no proline, or if there is any other amino acid except that of proline after arginine, then the digestion takes place. So that's how trypsin works. It will form different peptides, and those samples are then used in MALDI-TOF scan. So how exactly this, once you took this sample, what all steps you need to follow in order to, how exactly this digestion is done in terms of, in detail we'll discuss. So as you know, that specific gel has been stained. So now you need to destain that first. So the destaining is done using ammonium bicarbonate and acetonitrile. ACN is acetonitrile. Then you will incubate it for at 37 for 10 minutes, and so once you do that, the gel will become colorless, and you will get the, then you can, and it will dissolve also. So once that is done, you need to do dehydration using acetonitrile. You can incubate. So this dehydration steps requires incubation of 5 minutes, and then again add the same solution and incubate for 10 minutes. So once that is done, now you need to reduce the protein which is obtained. Then reduction is done using DTT. This is the similar preparation which we did for two-dimensional electrophoresis initially. Then we have run the gel. Similarly, here you are just in the proteins, you are just adding the dyes, right? And so similar kind of preparation here it is done before going towards mass spec. So now we will use DTT for digestion, then you will do alkylation. So there is reduction, then there is alkylation. For alkylation, you add iodoacetamide. So reduction is basically it will cleave the disulfide bonds. Here the disulfide bonds are attached with certain groups so that uh, oxidation is prevented. So those disulfide bonds once reduced, they will not come towards each other again. And once that is done, you need to dehydrate it again. And after that, you will remove the residual acetonitrile by incubating it for 5 minutes at 37. Then you will do the trypsin digestion, which you are talking about. Then you will add trypsin, and whatever proteins are present, they will be digested at arginine and lysine residues. Basically, cleave these disulfide bonds, and alkylation will add a group iodoacetamide group so that this oxidation doesn't happen. Again, they are not bind to each other. And the trypsin digestion, as we discussed, so if there is a proline, there is no digestion happening, and if there is no proline, that's how the digestion happens, and you will get these many number of peptides. So basically, what acetonitrile, it helps in dehydration of gel pieces and stain removal. Ammonium carbonate provides an optimum pH for trypsin enzyme to act, and overnight enzymatic digestion is done with trypsin, which cleaves after arginine and lysine residues at the C terminal and not the N terminal. So the a typical proteomic experiment. So this is what the workflow which we are talking about. And once then in the next lecture, sir continued about the same technique ahead. Again, gave an overview of that what we were discussing. So here pictorially you can understand what happens. So once you so initially so as we were talking about de-staining technique, so using acetonitrile and ammonium carbonate, so the stained gel will become colorless when you use reduction using DDT. So this disulfide bond breaks, and when you are doing alkylation, so this specific sulfide bonds will be attached to uh, acetamide groups, and that's how it will prevent the oxidation of the group. Then you will do trypsin digestion. It will after so this is N terminal, this is T terminal. So after arginine, this specific bond will broke, this will break, this will break after lysine. So if there is no proline after that, all this will be digested, and you will get these fragments for this specific protein. Then once you have that, as we discussed, you need to remove the salts and whatever present. That removal of salts happens with the help of this zig tip, wherein so your sample goes through it and the salts are removed and only peptides are present. So this is how the zip tip looks. So once you have the indigested protein samples after the processing is done, so you'll be using zip tip pipette tips containing C18 or C4 media. So it will have C18 or C4 media for enrichment of peptides, and salts and infringing agents, detergents are washed, and finally samples are eluted in a very small volume of solvent. So that's what this zip tip helps in. So this will also help you to remove all the salts which are present. So this is how it looks. So this is what the sample preparation whole MALDI technique requires. So you require matrix selection. You do the matrix preparation. You will do the sample purification initially, that is in gel digestion. Once you have the sample, so you'll mix the matrix as well as sample together. Once you mix that, then you will deposit those samples on the plate. Then matrix after deposition on the plate, you will dry them. You need to dry the plate, and once you dry the plate, then you will add that specific plate into the MALDI-TOF instrument and do the analysis. So as we were talking about different matrix, so there are different matrix available, like alpha cyano 4 hydroxy cinnamic acid, like which is called as alpha cyano or synapinic acid. So each type of matrix is used for certain purpose. So there are certain other DHB, THAP, picolinic acid. These are certain matrix, other matrix as well. So each matrix is used has a certain purpose. For example, your protein is such a way that your peptides are generally less than 5,000 daltons or lipids or nucleic acid. Then you can use this specific matrix. Similarly, if they are having higher than 5000 Dalton, then you use synapinic acid. Similarly, if there are small molecules and peptides which are not ionized by other matrices, you need to use DHB, which will help you to which will help for ionization. And similarly, if there are phosphorylated proteins, you need to use THAP, which is trihydroxyacetophenone if you are dealing with phosphorylated proteins. And for nucleotides, picolinic acid is used. So this is how the plate looks, 196 well plate, and this is how small volume you require for understanding, for doing the proteomic analysis using MALDI-TOF or determining that specific protein. So matrix preparation is done by mixing uh, matrix into a suitable solvent and vortex for few minutes to dissolve it properly. So once you do that, add the sample and as well as matrix together. So these are certain matrix. Accordingly, according to your need, you will use that specific matrix and add it onto the plate. So this is how the plate looks. Then once you add, so this is how the software uh, looks like. You will accordingly choose what all spots you want to analyze wherever you added your sample, and then you can click on fire in order to. So for example, this spot is chosen. That spot will be ionized first. So basically laser will come in contact with that. These samples will be ionized and it will run towards the top and then towards the detector. So this sir gave us one of the example of one of the protein, which is BSA, which is bovine serum albumin. So how this is how the spectrum looks in terms of this specific protein. So there are these are certain peptides which are generated after trypsin digestion, and this is how it looks. So MALDI can be coupled to tandem TOF or hybrid QTOF as well. So in order to make it more accurate or more accurately you want to analyze, you can add one more analyzer like so MALDI-TOF. So your TOF is one analyzer. If you add one more analyzer, that is called as other TOF. It's called as TOF. So MALDI can be coupled with another TOFs as well, or else you can make it a hybrid as well, like QTOF and TOF, and separated by a, so it is separated by a collision cell basically, and so it will help you to give much higher sensitivity than triple quad technique or single TOF technique. So these are basically different analyzers, okay? And we are majorly dealing with TOF technique itself. So whatever TOF was doing here, it will again do. So basically, it will again break this. So if this is the one specific ion, so this collision cell will again break this ion and it will separate that. So the analysis becomes much more accurate, and you will try and determine it in a accurate way. So whatever peaks you are getting, there is one of the software which helps you to understand what peaks that might be, which is called as mascot peptide mass fingerprint. So this specific software will help you to understand what all peaks are present and what they are predicting. So you need to do certain whatever you have used in your experiment, like you use trypsin for digestion. So all those details you need to give first, and once you give it, then this is how the analysis will give you. If there is an error, it will be red in color, and you will, if you are not given certain details properly, then it will tell you to add those details. Then this is how the certain proteins or certain peptides which are present, you can accordingly check those specific proteins, and the peptide sequence is also given accordingly. So basically, a query sequence is also given. It will accordingly compare with that query sequence. So whatever you want to analyze, if that protein is present or not, you will add the sequence of that protein here in the query sequence, and it will try and compare the peptides which query will form. Like if a query protein sequence is digested by trypsin, what all peptides it will form, and if your sample analysis is giving you the same peptides or not. So that's what it will compare. Then if the comparison is higher, that will give the higher score, and it will tell you. So basically, it is better the hit, and it will help you to uh, determine the protein more accurately. So the hit should have at least two peptides, and higher the sequence coverage, better the hit, and Y and B ion should be matched with MS spectrum. Then Sir talked about certain challenges involved in it, like translational proteomics research is a broad field that aims to identify novel diagnostic and prognostic markers, which, as Sir gave one of the example of prostate cancer. It also focuses on identification of certain novel therapeutic drugs. So these are certain various ways by which you can use Deitch technique and mass spec technique. So basically, proteomic studies which help you to do all of this. It can have a long-term objective as well as to provide personalized medication therapy and as well as translation of knowledge obtained from basic proteomics research uh, is a challenging task. So, but there is a huge gap between the bench side research and the bedside implication. So, currently, whatever we are trying to do is a bench side research in the lab itself. And once the biomarker is obtained, it is difficult to go to till the bedside implication in indicating that. So, people will do those tests itself, like whatever biomarker you predicted. People will talk about those specific. Biomarkers only and understand whether it is progressed or not. So basically to do that, you'll actually need a very high number of samples so that your research becomes much more accurate. And it's often making it difficult to translate findings obtained under lab environments in actual applications because of the same thing. One of the reason might be uh, many number of samples you need to analyze to come to a one specific conclusion. Then identification of potential biomarkers can have two approaches, prospective and retrospective. So backward approach can be like, which is retrospective, can be like target identification, biomarker validation, which we were talking about. And then if you validate that this is a one specific protein which can upregulate in certain disease condition, then therapeutic response to those specific subgroups, you'll try and treat them and understand that. Then forward approach can be the prospective clinical trial, like patient stratification in the sense dividing those patients, and then you will select an optimal drug in order to uh, treat them, and then whatever the patient benefit, that's the forward approach. So you can, this proteomic study can use in two different ways. It can be either backward approach or the forward approach. So backward approach, which is something which we were so the person is already having the disease, and we are trying to compare with the control and the diseased one and trying to check which are certain proteins which are upregulated or downregulated. That's the backward approach, which one of the examples are gave. But that you can also perceive this proteomics research in a forward approach as well. For example, the disease patient, and that the if you are giving certain drugs to certain patients, and if they are recovering from the disease, then accordingly that specific drug will selection of optimal drug basically, and that will help you to understand this specific drug can be given, and it actually helps the treatment approach much more accurately, and it will reduce the or remove that specific disease and such kind of stuff. So it can have either prospective study or retrospective study. The one example which Sir gave was a retrospective one. So, but still there are certain challenges associated with the translation, that is from bedside to the from the bench side to the bedside. So it is lack of standard protocols because everyone is following their own specific protocols, and it cannot amplify proteins like PCR because PCR helps you to gene amplification, but we do not have any technique which amplifies proteins. Then the presence of protein isoforms, huge variation in proteome with time within a same individual, because the proteome generally changes within an individual per day actually, and a broad dynamic range of protein concentration and complexity of biological fluids. So these are certain challenges basically, because the biological fluid is itself so complex, and so there is a range of proteins also. So detecting one specific protein, and if you are doing the same kind of sample preparation, same individual, at one specific time he might have a different pool of proteins, and another specific time he might have a different pool of proteins. So it is very difficult to replicate also such kind of uh, analysis, and variations among individuals as of same or different population, difficult to establish standard biomarkers applicable for all population. So that's what certain challenges we are having in terms of this. But still, we initially discussed about n number of benefits using dice technique and MALDI-TOF, but for applying it towards the bedside, these are certain challenges basically. So in the whole lecture, so sample preparation was discussed, how MALDI-TOF is performed, how TOF can be added for making it more accurate, MSMS analysis, and an overview of challenges associated with it. So these are certain references which sir has provided. You all can go through these certain books which will help you to understand in a much more deeper way of how mass spec is performed. So this is it from my side, and we can we can take a break of 5 minutes or so, and then we can discuss certain questions related to the topics we discussed today. Okay. Okay, Aditi will discuss questions related to today's topic. Try and answer them. Just oh, yeah, this will be better. Okay, so this is the question. What is the advantage of 2D dyes over 2D gels? What do you think? So yeah, more than one sample we can run. That is one of the benefit. Are there less running artifacts? Are there reduced gel to gel variations? Do you think the all three are the advantages, or only one is the advantage? So what is the answer? Like, is it D, A, or C? What do you think? Answer is A. Yeah, yeah, it can run more than one sample. That is true. Don't you think it has less running artifacts? Don't you think it has reduced gel to gel variation? Because D option says all of the above. Yes, we did talk about it, right? It has reduced gel to gel variations. It has less running. So reduced gel to gel variation, it is similar to that of less running artifacts, right? So the answer is all of the above. Yeah, we were discussing about the protocol, selecting the correct order of steps to be followed during in-gel digestion of proteins. So we did discuss in detail about how in-gel digestion is done. So once you cut the one specific spot after running the 2D like after running the dye experiment, you'll cut the gel. What you will do? The first step, you will do dehydration, destaining. So you'll obviously do destaining first. So it can be either A or B. So one after doing destaining, what do we do? We do dehydration first, then reduction, alkylation, then again dehydration and digestion, or we do reduction first, alkylation, and then dehydration, then digestion, or and then dehydration again after digestion. So as I said, as you know, like obviously, like the first step is obviously destaining. Yeah, the answer is A. Destaining, then we'll do dehydration, then reduction, alkylation, dehydration, and digestion. Yeah, right? Okay. So now you have samples prepared for protein and you want to analyze using MALDI-TOF. The sample has high abundance of phosphorylated proteins. Okay. So we did discuss like each and every matrix. So your most suitable matrix you want to identify. As I said, each specific according to the reason which specific matrix you need to use. So here the reason is you have phosphorylated protein. So which one was that? We discussed in the lecture. Is it synapinic, picolinic, 2,5-dihydrobenzoic, or trihydrosyacetone? Do you remember? Or should we go toward those slides? So the question is for phosphorylated proteins. Mm, okay. So phosphorylated. Okay, this is the one. So it is trihydroxy acetophenone. The answer is D. So the answer is D here. Yeah. So we were talking about side dyes as well. It forms covalent bond with the amino group of a particular amino acid. Which of the following is correct? So we discussed the how side dyes are labeled. So does it bind to methionine, cysteine, lysine, or arginine? Which is that amino acid side is bind to and make a covalent bond? And it binds with the amino group as well. So we did discuss about this in the lecture. Which is that amino acid to which side is bind? Yes, it is lysine. Okay. So which of the following enzyme can be used for proteolytic digestion? Trimotrypsin, trypsin, pepsin, or all of the above? All of the above. Okay, Mrunalini. C, B. Yeah, we did. We in the experiment or the in the lecture which we were talking about, we talked about only trypsin. But along with trypsin, we can also use chymotrypsin or pepsin. We can use any of those proteolytic enzymes, and each and every enzymes behave differently. So depending upon your protein of interest, if you know that your protein has more number of arginine and lysine, and accordingly you can use trypsin. But if you think your protein has accordingly, so these two work in a different way. So according to that, you can use any of the proteolytic digestion and use that. So all of the above is the answer and not trypsin only. Although in the lecture we took an example of trypsin digestion, but we can use any of them. Okay. So now in MALDI-TOF analysis, the analyte is mixed with the matrix and spotted on the MALDI plate. Match the following matrices with their suitable sample type. Okay, so these are the options. Yeah. So the first one, synapenic acid. So we just now in the third question said that this is for phosphorylated, but there's no option for 5. Is it small molecule, small nucleotide? So we know that for small nucleotides, which one is used? So if you go through this again, for small nucleotides, we use picolinic acid. And for small molecules, we use DHB, right? This is also for small nucleotides, the trihydroxyacetophenone. And generally used for nucleotides is picolinic acid. So as trihydroxyacetophenone is for nucleotides, picolinic acid is for nucleotides. So 5 is 3 and 4 is 5. So 5, 3 has only one option here. All others are having 5, 4, 5, 2. So the answer should be A, that is the first one, 5, 3. So what do you think? The first is 4. So this one is generally for peptides. The 2,5-dihydroxybenzoic acid is for small molecules. Synapenic acid is for proteins bigger than 5 kilodalton. Picolinic acid matrix is used for small nucleotides, whereas this is used for generally nucleotides, and whereas TH acetophenone, trihydroxyacetophenone is used for small nucleotides. So you'll have to remember them. So these are different matrices, and depending upon the protein of interest, different matrices are used in multiplate. So you can go through these so that if you remember these, it is easier to answer. Yeah. Okay, let's do the next one. So after the control treatment, protein samples are labeled. Which of the following reagent is used to quench the unreacted dye? We did discuss that. So in 10 millimolar of one of the reagent is used so that cyanine dye, whatever is the extra dye left, whatever was supposed to be reacted, it's reacted now. Whatever is the extra left, the unreacted dye. So you add something so that quenching becomes complete. So is it lysine, cysteine, urea, or SDS? Yes. Yes, Aditi and Mrunalini. Aditi, you were present for the whole lecture. I think you should be able to answer. And Mrunalini, if you have gone through sir's lecture, even you will be able to answer the question. It's easy. Yeah, what do you all think? You can just guess if you are not understanding the, like, you are not able to get the answer. You can still guess it. Okay, okay. So why did you come at 3 o'clock? Our lecture had started at 3, right? Occupied? Okay, okay, fine, fine, not a problem. I'll any which ways will add it on the YouTube, like for your the recorded lecture. You can accordingly go through that as well. Yeah, so that it becomes like it will be a revision for you all. Sir has already explained you in a broader way. I've just revised the whole 5 lectures again, the week 4 5 lectures. You can go through it again. So the answer is lysine basically. So the cyanine dyes basically bind with the lysine residues, which we discussed here itself in this question. So cyanide, the particular amino acid it binds to, its lysine. So once all the lysines are occupied of the sample protein, whatever are the remaining dye, unreacted dye left, you will add more amount of lysine to it so that those unreacted dye also binds to the lysine. So each all the component of the cyanine dye is reacted with one of the lysine, although the lysine which we added or the lysine which is present in the sample. Okay. So which of the following parameter is used to create an identical spot boundary across all channels in Dyes data analysis? So this is the soft mass spec analysis. This is the biological variation analysis. So we want to create an identical spot boundary across all channels. So this is a little difficult question. So is it a detection or code detection? And which one will following parameter used to create channels in Dyes data analysis? So all the channels in the sense all different samples, and that will be can be done only via code detection, whereas these are different approaches and which we cannot. This will so as here the question is about creating an identical cross boundary. So so when you do so there is one sample, there is then only a spot boundary can be created. I think if I have a image of it, it will be easier to understand. Spot boundary indicating. So for example, so you need to do code detection basically. So for this protein as well as so as we can see this one protein at Psi5 die with Psi2 die as well as with Psi3 die. So all three will be overlapped with each other. So you need to do code detection basically, and then you can create a boundary towards it. Boundary in the sense you will make that this particular zone as a protein in all three samples. So, so if you want to create a spot detection thing, create an identical spot boundary, you can only do via code detection. That's what the question is. So, as here the question says about creating a spot boundary, so that's how you can create it. Then we did discuss about zip tips as well. Having C18 columns are used for sample enrichment, basically a peptide enrichment and salt removal. And sample containing digested proteins binds to the C18 column matrix. Which of the following biological interaction happens with that C18 column? Is it ionic interaction? Is it covalent bond interaction, hydrogen bond interaction, or hydrophobic interaction? So if I just, it can be either ionic hydrogen or hydrophobic, but it cannot be covalent, because if you think C18 column matrix, if it is covalently attached, then you will not be able to elute that specific protein and add it in the MALDI plate. So that's how this zip 18 or zip tip looks. So if you see, we just talked about it. So how does zip tip looks? So this is how zip tip looks. So inside that there is C18 matrix, and if the binding is happening covalently, then it will not never be eluted out. The peptides will never be eluted out. It is covalently bound means it is strongly bound. So obviously B cannot be the answer, but anyone can be the answer. But what do you? Yes, yeah, that is the answer. So that's how the hydrophobic. So I was trying to just eliminate the other option so that it will be easier to come to one conclusion. So yeah, the answer is as Mrunalini directly said it. So if it is C18, it will be probably hydrophobic interaction. That's what the answer is. So your extracted protein sample has high salt contamination. So it says that high salt contamination is present, but you do not have zip tips for sample cleanup. So zip tips are generally used for cleanup. You are removing salt, you are enriching your samples. So that's what zip tip is used for. But now the question says you do not have zip tips for sample cleanup. So which of the ionization technique you will use? So as your sample is contaminated with high salt, so now you need to deal with come up with certain ionization technique which can tolerate high salt contamination and would be preferred to analyze your sample. Is it electron spray ionization? Is it MALDI? Is it electron ionization or none of the above? We did talk about one of the technique which has high salt, which can tolerate high salt in terms of ionization technique. No, it is MALDI itself. So MALDI, yeah. So MALDI has a potential to tolerate high salt as well. So even if there is basically electron spray ionization cannot be used neither. So these techniques are sensitive to salt, whereas MALDI is not sensitive to salt and it can tolerate high salt contamination. So the answer is MALDI. Yeah. So if you can see here in terms of MALDI advantages, which we talked, that is MALDI here. Just a minute. Yeah. So it is more tolerant to salts than that of ESI, and this so that's what the advantage of MALDI is. So even if your sample is contaminated with salt, you can still go with MALDI. Yeah. Okay. So which of the following is advantage of Dietz technique over gel-free approaches? Is it more sensitive, detects modification of intact undigested proteins, allows greater range of proteins to be analyzed? What do you think all of the above? Aditi, what do you think? So the question is advantage of Dij over gel-free approaches and not the other way around. So gel-based technique, which is difference in gel electrophoresis over gel-free approaches. The answer is not all of the above. So as that's the reason I mentioned that it is not the other way around. Gel-free approaches are generally more sensitive. So it detects, yeah. So the question was other way around. Yeah. So gel based will actually help you to even detect the intact undigested proteins also, whereas the gel free, you need to digest those proteins and then only you will be able to analyze, or else you won't be able to do that. So yeah, yeah. Yeah, but it's not that sensitive, so yeah. And getting greater range of proteins, I think there are more gel-free approaches methods available. In terms of gel-based, there is only 2D gel electrophoresis and dyes majorly used in terms of proteomics. But there are n number of different gel-free approaches available, so that becomes that will help you to have a greater range of proteins comparatively. Yeah. So which of the following criteria is used to select the matrix? We just discussed in terms of MALDI-TOF. Is it the charge on the sample? Is it the molecular weight and nature of the sample or isoelectric point of the sample? Or all of the above? I think we are left with only next 10 minutes, so I'll be a little quicker. So it is the nature of the sample as well as molecular weight. So that will help you to understand which matrix you need to select. As we were just discussing these different matrices. So these are different matrices available and they are decided on the basis of the nature itself, you can see, as well as the molecular weight or else the nature, whether they are small, they are nucleotides, whatever it is. So depending upon the molecular weight and nature, you will determine the matrix to be used. So that's what the question was. Which of the following criteria? So it is molecular weight and nature of the sample. So which of the following should be minimized any biasness in sample processing in dye experiments? Is it dye swapping, addition of cysteine, use of sci-fi dye, or all of the above? So we did discuss in detail about sample preparation in dye experiments. Aditi, are you there? Are you understanding? You were there for the whole lecture, but you are not able to answer the questions. Are you facing any problems or you are getting it but just unable to answer right now, or you're not getting it at all? Yes, Aditi, are you there? Okay, so what do you think? Say, we want to minimize any biasness here. So which of the following should be minimized? Should be used basically to minimize biasness. Yes, you are here, but you are not answering the questions as you were there for the whole lecture also. Is it becoming, do you think the questions are a little difficult to answer? Yes. Bit difficult, yeah, but like once you go through the lecture again, maybe it will be easier for you to answer. As it was just the first time, it might be becoming a little difficult. I agree to that, but yeah, you can go through it once again, it will be easier for you to answer. So this is dice swapping you should avoid because obviously if you swap the dice, you are creating certain biasness to it. So dice swapping must should be used to minimize any biasness. Spectrometric technique involves embedding of the sample in a crystalline matrix and then desorbing it with a laser of ionization. So it is in the question itself, you can directly answer it. So then desorbing it with the laser, it is in the question, but it itself it is MALDI. So this was not that difficult to answer. Yeah. So in two-dimensional electrophoresis, which dimension separates proteins based on the isoelectric point. This is also easy to answer. Which is the first dimension? Oh, sorry. Yeah, it's the first dimension, the isoelectric point. What is the second dimension? Yeah. uh, Okay. So the mass spectrometry could be used for protein identification, protein characterization, protein quantification, or all of the above. A little tricky question. Yeah, it is all of the above. Yeah. Yeah. So identification for sure, but you can characterize it as well because that's yeah. So that's why you can be able to characterize as well and quantify if you have a standard. If you have the standard, then it will be quantifiable as well. Yeah. So which of the following organic solvent is used as a mobile phase in liquid chromatography based experiment? Yeah, generally that's what is used. I think you are already doing certain research related to it. That I think so. Okay. 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 Yeah. So which of the following is the correct sequence of events in case of mass spec? Yeah, I think even this is a bit easier to answer. So obviously the first step should be, you all can answer the question. Yeah. So obviously the last step will be detection. It can be either these three out of these three. Is it a first step is acceleration or ionization? Then you will be able to answer the question. Yeah. Yeah. Yes. And then the deflection because if you have the mirror and then it goes towards the detection detector. So in which of the following mass spec technique the sample is introduced in solution form, which is eventually nebulized under an applied electric potential? Oh, okay. So this is the key word here, I think, nebulized. If you all know different ionization technique, it will be easier to answer, and if not, then it will be a little tricky to answer. So in electron spray ionize, yeah. So if it is, so that's what I said. So this is the word which will help you to answer the question. So it's electron spray ionization. So it has a nebulizer basically in that ionization source, and that's what is used for in electron ionization. The path of ions after deflection depends upon. So what? So once the deflection happens, what is the path of ions? Is it only on the mass of the ion, only the charge, or both charge and mass of the ion? Yeah. So generally, mass spec works with mass to charge ratio. So it depends upon both mass as well as charge. So depending upon mass to charge ratio, that's how the ions travel towards the detector, depending upon both. Yeah. So that's it. I had 20 questions. So this I'll also add this specific PDF to the link as well as the solutions of it and as well as the PPT which we discussed and as well as the recording of today's lecture in YouTube. So you all can go through that. So this is it from me. And if I get, sometimes NPTEL gives us an opportunity to take one more lecture by just clearing the doubt session if people wanted, as well as if we get a chance to do that one lecture, we might do that, or else this is the last lecture. Yeah, let's okay. 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 Yeah, yeah, yeah. Good. Yes, yes, thanks. So, where are you from? Like, you are teaching somewhere, or you are doing pursuing research? Oh, okay. Nice, nice. Nice to talk to you. Oh, okay. Familiar with. Okay, okay. Oh, okay. Yeah, there are many. I think if you will go through week one or week two lectures, I think the initial introductory part it had given a broad overview of gel-free approaches. Yeah. Oh, okay. Nice, nice. Yeah, nice to meet you and nice to talk to you. Okay. Yeah, Aditi, even thank you. Thank you for joining. Thank you for attending the whole lecture. If you people have any kind of doubts, you all can, I think you people have my email IDs as well. You can contact me if you all want. If you want to contact me, I'm just writing the email ID of mine. 214021626016 @itb.ac.in. You all can email me to my email ID. I think email is being given to you all if I'm not wrong. Like here, you all have this particular sheet with you all. If not, I think you all are having. Where is the other sheet? Okay, I'm not. So you all can access this specific folder as well. I have added all those week's presentations as well as questions. You all can go through it. I think the link is being provided to you all as well as you all can go through this YouTube lectures as well. I've added week one, the proteomics based lectures. You all can go through that as well. Yeah. Okay, thank you. Thank you for attending. So if you all have any kind of doubts, you all can email me. I can go through that and accordingly answer you all. Thanks. Thank you. Thanks, Aditi. Bye bye.